Hello students, welcome to E-Patashala. I am K. Muralidhar from South Asian University, where I am a J.C. Bose National Fellow and an honorary professor. Today we are going to talk about structural basis of living, a paradigm in biology, from the paper Introductory Biology. What do we learn from this module? The learning outcomes at the end of this module, one will be able to realize with deep reflection that all biological, that is living activities, are emergent properties. Appreciate that there is a hierarchy of structures into which living processes are expressed. Understand that the structural integrity at each level, that is population, organismic, cellular, organelle and molecular, appears to be essential for the respective living activity. Appreciate the obsession of a reductionist biologist to relate structure to function. Realize that as far as emergent properties are concerned, that is evolution, migration, reproduction, secretion, defense, response, development, communications, movement, enzyme activity, pharmacological activity, etc., a one-to-one structure-function relation is hard to find. Interactions among components leads to emergent properties. Realize that systems biology approach is the only way to understand the mystery of biological phenomena like movement or memory, emotion or thought or many others. Let us introduce this subject a little more. Biology is the science of life, including living organisms and living activities. In Module 1, we grappled with the idea of living state. In Module 2, we were left wondering at biodiversity. In Module 3 and 4, we surveyed what all the organisms do. Activities like growth and reproduction, development and differentiation, movement and transport, homeostasis, language and music, discrimination of non-self from self, the extraordinary biocatalysis, hormones, signaling molecules, transporters, contractile proteins, and excitable membranes, electrical conduction, etc. mystify us with regard to mechanistic details. Biologists are obsessed with the idea of establishing cause and effect relationship in every activity. While the reductionist biologists look for proximate causes, evolutionary biologists look for distal and ultimate causes. However, the truth is all activities are at all levels of organization are stochastic processes. They are more dictated by probability than mechanistic causes. We all realize that almost all the activities at the molecular level are mediated by functional proteins. The hereditary material, on the other hand, appears to be a nucleic acid. There is either a physical or a chemical basis of all activities. In the case of some activities like memory or emotional experience, we are not sure of what structures are the basis of these. In this module, let us try to understand the structures of ecosystems, populations, organisms, cells, subcellular organelles, and less complex macromolecular aggregates, macromolecules like proteins, nucleic acids, polysaccharides, and small molecules like amino acids, monosaccharides, fatty acids, secondary metabolites like alkaloids, terpenoids, etc., and try to prove that structure subserves function in biology. However, let us also remember that function, or better called activity, need not always be physiologically relevant. Therefore, toxins and drugs from nature are also included. In this lesson, we asked a major question: how are living organisms structurally organized? To get answers, we can look at it at different levels of hierarchy of our structural organization, starting from molecules all the way to ecosystem. So if you begin at the molecular level, we have already learned that all living organisms are basically made up of biomolecules, which is a euphemistic name for chemicals. And these biomolecules are classified into two major types. One called small molecules, otherwise also called as biomolecules. and the other are macromolecules or big molecules. And the distinction is that all small molecules or biomolecules of molecular size less than 800, and all macromolecules have molecular size above 10,000. In this slide, there are indication of a representative biomolecule, small molecules like glucose, which is a carbohydrate, and you also see ascorbic acid, otherwise called as vitamin C. You also see amino acids and fatty acids represented at their molecular form. These biomolecules are classified into approximately 30 to 50 subgroups. One of them represents what we call as amino acids. Another group represents sugars. Another group represents heterocyclic compounds. What is important to understand is many of these compounds, simple molecules, have stereochemistry behind them. Here is one example, an amino acid called aspartic acid, and as we learn from the structure of this, it is represented in what is called as a L form, capital L. It has nothing to do with optical rotation, it simply means a configuration, one of the configurations. Similarly, there is a D-aspartic acid also, but which is rarely found in proteins or living systems. Therefore, many of these compounds have stereochemistry and particular configurations only are available or occurring in living systems. Amino acids are always found as L, except in some bacteria where you will see D amino acids. Similarly, sugars are always found as D, in very rarely, like in some cases like ascorbic acid or gulonic acid, you will see L. Otherwise, they are always D. Here is an example of a tertiary structure of a representative hormone called prolactin. This is a hormone. It is a polypeptide of approximately 200 amino acids long, but as you notice here, it has twisted and contorted in the form of different helices. There are 4 of them depicted here, and you also find between the helices where there is no structure, which we call as loop. Together, the overall shape of prolactin is indicated here. This is called so-called tertiary structure. Biological activity like enzymatic activity, antibody activity, hormonal activity, receptor activity, any pharmacological activity or physiological activity is essentially totally dependent on integrity of tertiary structure. Any disturbance to this tertiary structure in the form of partial or total denaturation results in loss of biological activity. All our physiological functions are mediated by functional proteins, and therefore it is extremely important to relate this structure level to the mediation of its characteristic physiological activity. Here is another example of the so-called biomolecules, one of the major groups of biomolecules extremely important in our body functions, and these represent a class called steroids. Most of you have heard of a major sterol called cholesterol, of which there is a lot of discussion in biomedical research because it has something to do with our cardiovascular disorders. But in this particular slide, you see examples of sex steroid hormones. That means hormones both in the female and in the male. Example given is of one estradiol 17 beta and the other testosterone. These are steroid derived molecules but which have lot of biological importance as they function as sex hormones. On the right side also you will see another type of steroid hormone called as corticosteroids. In our adrenal gland, which is above our kidney, this corticosteroids are produced, and as you have learnt in general physiology, they have tremendous influence on our immune system as well as on our behavior. We have already learned that many of the biomolecules have a property called optical activity. How do we understand the structural basis of this? Look at the picture. You see here represented chiral molecules like a D amino acid, L amino acid. Long back in the history, scientists were looking at the crystals of glyceraldehyde, and they found that physically they could separate by noticing some of these crystals which were looking on the right side and some which were left faced. They called these as respectively D and L. It so happened that subsequently, when people analyzed the structural basis of this optical activity, they could arrive at the same similar structure present in all such chiral molecules. Some of them are left facing, some of them are right facing, and therefore all chiral molecules like L-amino acids, D-glucose and other sugars are essentially described in terms of describing D-glyceraldehyde and L-glyceraldehyde. This was based on that there are 4 substitutions different on a single carbon. This was one level of analysis, but subsequently people showed that there is chirality in certain molecules where you don't see an asymmetric carbon, but overall the shape will exhibit this chirality. Like if you take a alpha helix of a protein part or similar structures, we have chirality here, not based on what we learnt just now, that there is an asymmetric carbon with 4 different substitutions, but the overall shape of that is chiral. Hence, chirality is a property of some biomolecules, and most of the molecular recognition that underlies physiological processes is essentially due to this chiral molecules playing their role. Therefore, one should give more importance to this property of chirality and understand how much role it plays in physiological recognitions. So at the next level of hierarchy, how these small biomolecules assemble themselves in a particular way to become macromolecules or molecules whose molecular size is above 10,000 Daltons. Here this slide gives you some examples. For example, if you take a macromolecule like protein, it is made of monomeric building blocks called amino acids. So we have already seen some of the amino acids listed. These amino acids are joined together by a characteristic chemical bond called peptide bond, and that results in a polypeptide or a protein if it has a biological function. Similarly, if you take a polysaccharide, of which many examples can be given, one is talked about here, starch, which you get from plants. It is made up of monosaccharides or monomeric building blocks called glucose. It is polymerized, and the linkage that you see in a polysaccharide is called a glycosidic bond, just like a peptide bond characteristic of polypeptides or proteins. Similarly, another example is given. Nucleic acids, which are all genetic material, they are made up of nucleotides, and the kind of bond that links the nucleotides into nucleic acid form is called a phosphodiester bond. Now, therefore, what we learn is that monomeric units join together in characteristic chemical bonds to form macromolecules. However, if you look at all the macromolecules available, they can be classified into 2 types. Those who are got one type of monomeric building block which has polymerized. For example, if glucose is joined by another glucose and this goes on hundreds of residues, you get what is known as a homopolymer of glucose, otherwise starch or cellulose. Similarly, you have others where the monomeric building blocks comprise more than one type, then you get heteropolymer. Nucleic acid is a heteropolymer. Peptide or polypeptide or proteins are heteropolymers. But you also have simple homopolymer version of these things like poly T or poly A, or in the case of polypeptide, say polyglycine or polytyrosine or polyaspartic acid. Similarly, in the polysaccharide, we have homopolysaccharides and heteropolysaccharide. That is what is depicted here. When we look at these polymers, homopolymer or heteropolymer, and discuss them in little more detail, we notice that the word structure has different connotations when we talk about polymers as opposed to when we talk about small molecules. As an example, if an inorganic chemist talks about the structure of sodium chloride, all he will do is he will write sodium and chloride ion on the board as a biatomic molecule. This is basically its molecular formula. On the other hand, if you talk to an organic chemist and ask him to describe him or her the structure of benzene, he will write a two-dimensional diagram. But when you talk about polymers and ask a biologist or a biochemist to write the structure, what you will see is four types of answers, because there are four types of structures. One is called primary structure, where all the building blocks are arranged in a particular sequence, and this sequence is important, and that constitutes primary structure, which results in a linear form. However, people have found by experiments that the end-to-end length of these linear forms is actually more by calculation but less by experimental determination. And the reason for that is that these linear forms assume certain shapes which results in secondary structure and tertiary structure. Examples are depicted here. In the case of proteins, the secondary structure can be an alpha helix or a beta pleated structure. Similarly, in the DNA, we notice what we call as the Watson-Crick model is essentially bDNA, or the structure of DNA at that hierarchical level. Beyond this, there is the backbone itself will fold itself into characteristic forms like a woolen ball, and that level of structure is called tertiary structure. Therefore, we should recall that polymers like proteins, nucleic acids, polysaccharides, especially nucleic acids and proteins, have their structure organized in a hierarchy, and this hierarchy from bottom to top essentially represents primary structure, secondary structure, tertiary structure, and quaternary structure. The primary structure is essentially how the monomeric building blocks are arranged in a linear sequence. The secondary structure essentially is how you can get end-to-end length shortened by bending of this backbone in characteristic forms. One is indicated here for a protein. You get what is known as alpha helix. And then at a higher hierarchy structure, we notice that even this secondary structure fiber is bent upon itself and undergoes a lot of contortions, giving you, stabilized by certain chemical bonds, you get the overall spherical shape, and that is called as tertiary structure, which is shown here for certain proteins like myoglobin. Now, if you look at another protein like hemoglobin, we notice that four myoglobin type of proteins are held together. That means it has four subunits, and now this structure is called a quaternary structure. In biological functions, in discharging physiological functions, primary structure, secondary structure, tertiary structure, quaternary structure are equally important. Now, the theme of this lesson is the structural organization of biological systems or living organisms. We began with small biomolecules. We then said that biomolecules, small biomolecules, some of them get assembled in the form of polymers called macromolecules. And these macromolecules, in their turn, essentially assemble or interact with each other and form characteristic structures which are microscopic in nature. Some of them are called a cell membrane or subcellular organelles like ribosomes or endoplasmic reticulum or nucleus or any such organelle. And therefore, for a biochemistry person, subcellular organelle level of structure is essentially described as macromolecular complexes. For example, a group of RNA and proteins molecules combined together in characteristic way of which we don't understand the mechanism into structures called ribosomes, which are involved in protein synthesis. Here is an indication of another such structure called cell membrane, which is popularly called as a lipid bilayer, following the Singer-Nicholson description that it is a fluid mosaic in nature. Here is depicted the two layers of the membrane, and in which we see characteristic integral proteins embedded, and also the lipid molecules or part of the phospholipid molecules indicated. On the right hand side, you will notice another macromolecular assembly, but this is an assembly between an enzyme, which is a macromolecule, and its characteristic substrate. What is indicated and what is learned from this slide is that the substrate, which is a small molecule, has to fit in a part of the structure, tertiary structure of the enzyme, a place called active site, which is a three-dimensional structure. And therefore, we always say that any interaction, molecular interaction between small molecule, big molecule, and big molecule, essentially resulting in macromolecular complexes, is a stereospecific recognition or molecular recognition. In the words of Linus Pauling, all physiological reactions are essentially molecular recognitions. This is an example where we are discussing the binding of oxygen to hemoglobin. Most of you know that hemoglobin is present in our erythrocytes, that it captures oxygen in our lungs through the inhaled air and then carries it through blood circulation and delivers to tissues. Now, if we want to understand this process, we have already mentioned about the quaternary structure of hemoglobin. Each of the subunits has a binding site for oxygen. So if somebody does an experiment where you are studying the interaction of oxygen with hemoglobin and 1/4 of hemoglobin, which is also found as an independent protein called myoglobin, and measure this oxyhemoglobin, oxymyoglobin as a complex and measure it as a function of increasing concentration of oxygen, you get curves like this depicted in this slide. On the right hand side, you have a rectangular hyperbola, which means that as you increase oxygen, amount bound to myoglobin also increases. Ultimately, it will reach saturation point. And in the left hand side, you notice that of a hemoglobin, which is not a rectangular hyperbola. It gives you a shape of sigmoidal shape. Each of these is extremely important in the discharge of its physiological function. While myoglobin cannot supply, it does not move even, it is a part of the muscle, it does not supply oxygen to tissues. Hemoglobin, because of this sigmoidal shape, has a capability of binding oxygen in the lungs, whereas it delivers oxygen in the peripheral tissues. So, to continue with our theme, structural organization of living systems, we already noticed how small molecules become macromolecules. Macromolecules assembles into macromolecular complexes. Some of them become subcellular organelles, and these macromolecules per se and macromolecular assemblies discharge or mediate physiological functions. We can look at other subcellular organelles like nucleus, which is shown in this case, as well as what is shown on the right side is an example from the plant system called chloroplast. Therefore, subcellular organelles like a chloroplast, which is otherwise called as a plastid, and nucleus, which is present characteristic of all eukaryotic organisms, are essentially macromolecular complexes. The description of their composition in terms of macromolecules, small molecules, and how these are assembled to form this subcellular organelle, which is a microscopic structure, is you have to go into detail about the kinetics and thermodynamics of these intermolecular interactions, which is a huge chapter in biology, and we don't have time to describe all this here. What we have learned till now is that molecules assemble into bigger molecules and bigger molecules assembled into subcellular organelles. And then a group of subcellular organelles enclosed by membrane essentially becomes a cell. At this level of structural organization, if you look at cells, then all organisms can be classified into two groups: unicellular organisms, which are whole body is made up of one cell, and multicellular organisms where the body is made up of more than one cell. If you look at all these cells, whether from unicellular organisms or multicellular organism, what strikes one is the variety of shapes they exhibit. There are cylindrical so-called cells, there are spherical cells, there are cells with flagella-like structures, and so on and so forth. There are a variety of shapes, and that asks, in the context of this lesson, how does a specific molecular interaction results in the shape of the cell, number one, and number two, how a given group of cells characteristically exhibit that shape and retain that shape every minute of their life as well as throughout generations. That essentially leads us to ask a biological question: how is cell shape regulated and maintained? Here are indications of on the left side, a bacterial cell, E. coli cell, which is approximately cylindrical, about 2 microns in width. Similarly, you have other examples of the so-called shell shape diversity. So when we talk about multicellular organisms where cells associate with similar cells and form into tissues and organ systems, we can notice that whether it is animal or plant, tissues contain cells, but they are organized in a particular shape and there is a particular intercellular interaction. We will take one example from plant systems. We notice that if you take a leaf, of which we have learnt in earlier lessons, this leaf has got pores on the leaf, and these pores are called as stomata, and these stomata are places where gas exchange takes place. You notice in this slide structure of a stomata which contains guard cells. These have characteristic kidney bean shape in them, and they close and open, and therefore the shape is altered in these kind of things. Similarly, if you look at other type of cells, there are what are called as parenchymal cells in the plant system, some of which are depicted in this place. What it shows is the important biological question: how different groups of cells recognize similar cells and organize themselves into what we call as tissues. Tissues are macroscopic structures. If you just want to look at a tissue, we don't need a microscope. But to look at cell shape, we need a microscope. So look at any tissue, you will understand the same principle. So students, let us now summarize what we have learned from this module. Living organisms are made of molecules big and small. They are also called as macromolecules and micromolecules. These biomolecules have structure. Biomacromolecular structure has 4 components: primary, secondary, tertiary and quaternary. These form a hierarchy. There is a certain degree of interdependence. Primary structure gives rise to secondary and tertiary and quaternary. In case of all functional macromolecules, secondary and tertiary and sometimes quaternary structural integrity is absolutely essential for exhibiting the function. Macromolecules interact with each other, forming nucleoprotein, lipoprotein and glycoprotein complexes. Chromatin is a nucleoprotein complex, while many hormones are glycoproteins. Lipids and proteins interact to form membranous structures. Unicellular organisms evolved into multicellular organisms. Biofilms are an intermediate stage. Multicellular organisms can have different types of cells. In still higher organisms, cells are organized into 2 or 3 germinal layers, ectoderm, endoderm, with or without mesoderm, each of which gives rise to characteristic tissues. In higher plants and animals, tissues are themselves organized into organs and organ systems. A mammal or a green plant is structurally very complex. The reproducibility of the characteristic shape and size of all the tissues and organs is a mystery in biology. Plant cells from an adult plant are totipotent. They can form any tissue or even whole plant in vitro. Animal cells, on the other hand, are not, with the exception of fertilized egg cell and embryonic stem cells, they are not totipotent. Cells of limited potency to differentiate into other cell types are present in bone marrow and umbilical cord. Every tissue has a certain number of stem cells who will, however, differentiate into only the same type of adult cell. A liver stem cell becoming a liver differentiated cell but not a blood cell. The formation of different tissues and organs from the fertilized cell is studied in developmental biology. What is not understood is how the shape and size are regulated. The study of development also includes how cells die. This can be extended to how organisms die. The organization of populations and communities and ecosystems and how those structural features support emergent properties at that level of hierarchy, say climate change or evolution, is a fascinating chapter in biology, but we know very little. The greatest challenge is to understand how an emergent property of brain like mind arises and how it is supported by structures.